Good afternoon, everyone. I'm really happy to be here, so thanks to the organizers for giving me this opportunity. As said, I will discuss the mutation landscape of phosphorylation signaling in cancer. And the true centerpiece of our work and many other labs is the TCGA pancancer analysis project that was published just a few weeks ago in Nature and Nature journals. And this was led by Josh Stewart, who's also in the audience here. For our own work, the major importance of this multidimensional data set is that there are more than 3,000 cancer genomes characterized in the context of somatic mutations. And as you can see from here, there are 12 types of cancer that are of major clinical and societal importance in the world. And what we would like to do with this type of data is to discover mechanisms of cancer biology, and more specifically, find which events are cancer drivers. So cancer drivers are somatic mutations in genes and pathways that create advantages to tumor cells. So cancer drivers, there could be different kinds. Some of them could be responsible for tumor initiation, others lead to tumor progression or help maintaining tumors, and ultimately lead to metastasis to different organs. And more often than not, those cancer drivers are discovered by high mutation frequency. So the central assumption is that if you see a tumor sample having a specific mutation and more tumor samples having exactly the same mutation over and over again, then you could assume that this is statistically very unlikely that those mutations coincide, and instead these mutations are probably involved in tumor biology. However, we do see that most of the mutations picked up by sequencing are rare across many tumors, and also we know that most mutations are thought to be passenger mutations. So they don't have a functional impact on cells, but they are rather coming along with a genomic instability that is inherent to cancers. So here we decided to take a little step back and think about complex interaction networks that we know are ubiquitous in biology. And we have previous literature that shows that complex interaction networks are somehow involved in determining phenotypes, and also that they are disturbed in cancer and of disease. And when we think about the central dogma of molecular biology, then the components of these interaction networks, such as proteins or RNAs, are actually encoded in the genome. And therefore, they are subject to genetic variability that modifies these proteins, and ultimately we believe that modifications on the level of DNA actually lead to alterations in cellular signaling networks. In order to test this idea, we focused on protein phosphorylation, which was just discussed in the context of the seminal work by Tony Pawson. Protein phosphorylation is a post-translational modification which involves kinases as rider proteins, phosphatases as eraser proteins, a number of different domains that act as reader proteins, and they all revolve around a central modification, which is adding or removing a phosphate group to S, T, or Y amino acids in protein sequences. We know that phosphorylation has a regulatory function in proteins. It can activate, degrade proteins, lead to differences in folding, or manipulate with protein-protein interactions. And phosphorylation is also central in cancer biology, because we know that many central cancer pathways have phosphorylation events involved, such as cell cycle. And it's also important in drug targeting, because kinase inhibitors are really a center of targeted drug development. So here's a pretty busy slide, which characterizes our analysis. Essentially, we integrated two types of high throughput data. On the one hand, we collected a large number of phosphosites from phosphoproteomic experiments. That's around nearly 90,000 sites in 10,000 human proteins, and these are all experimentally determined by large-scale screens. And on the other hand, we took advantage of the large data set of somatic mutations provided by the TCGA project, which is nearly 250,000 of missense mutations in many cancer samples, several cancer types, and so on. We defined something we call phosphorylation-associated single nucleotide variant, or PSNV. This is a protein coding missense mutation that occurs at a specific phosphosite or very close to a phosphosite. So we decided to choose a seven amino acid region around phosphosites because there is previous evidence that this is the area where kinases tend to recognize their sequence motifs. And using this type of analysis, we identified a large number of genes with phosphosite-related mutations, about 17,000 phosphosites in total, and more than 90% of tumors have at least one mutation of that kind. Using that collection of mutations as our data set, we carried out several types of analysis. First, we looked at global trends affecting these types of mutations. Second, we zoomed in on specific genes where mutations of phosphosites are prevalent. Then we used pathway interpretation to look at rare variants and also more frequent variants. And then finally, we looked at kinase-substrate interaction networks to get some sort of more mechanistic insights of how these mutations carry out their function. As first, we looked at the global trends of phosphosite-related mutations. And just as a background, when people look at genetic variants, very often they use multiple different tools for predicting the function of these variants. Tools such as polyphen or SIFT essentially look at the coding variant and use different metrics to decide whether this variant has an impact on protein function or not. And here we used 5 different tools of that kind in order to get an understanding of how important phosphosite mutations are. As you can see from the plot, we used a majority vote scheme in which each tool had 1 vote, and we tried to estimate which variants are neutral and which ones are harmful. So variants which are harmful have 0 or 1 votes from these different tools of being harmful. And on the other hand, a variant is more likely to be highly harmful for protein function if all tools predict it to be a harmful variant. The summary of this global trend is that phosphosite mutations tend to be more harmful to protein function on average as predicted by those different tools. And that kind of suggests that when people do mutation analysis of function, then interpretation of those variants in terms of signaling information may actually give a clearer picture of what the disease mutations could be about. Next, we focused on specific genes where mutations of signaling events are prevalent. And we used our previously developed tool Active Driver, which would look at mutations near signaling sites and predict a statistical score of how important the enrichment of mutations is. And this allows us to interpret known cancer drivers in the context of signaling and then find candidate cancer drivers where mutations are rare but very close to signaling sites. And this is a logistic regression model for which I will not go into much detail, but I'll just mention that it will pay special attention to sites where there are several post-translational modifications, and it will also consider protein disorder, which is an important confounding factor in analyzing protein mutations. So here's just a quick overview. I'm sure that you will not be able to recognize your favorite genes from that distance. We found 150 cancer genes where those mutations are significant in terms of signaling. And there are 26 highly recognized and well-known cancer genes in this list. This is a statistical enrichment, which kind of confirms that we find the signal that we want to find. But also, there's a large number of pan-cancer-only genes, which are not apparent in any single cancer type, but if you pull together all these 3,000 cancer genomes, you will find these genes as sort of borderline significant, or this is the way how we can interpret rare mutations. And also, there's a number of genes which we find only by using our method and no other commonly used mutation significance computation finds. So this shows that our method is a bit complementary to other approaches that people use to discover cancer genes. Here's one example which serves as a good positive control. This is beta-catenin. It's a Wnt-activated oncogene that is known to be active in several cancer types. And it's fairly well recognized that this oncogene becomes active when the phosphorylation is disrupted in the N-terminal sequence. And you can see here from the top chart, this is where the mutations of beta-catenin accumulate. There's more than 20 per site at some point. Then on the yellow-orange chart, you can see where the post-translation modifications occur in this protein. And finally, you can immediately see that most of the mutations accumulate near a phosphosite cluster in the beginning of the protein, which is also shown by red. And this is statistically significant according to our model, and kind of confirms previous understanding of how this oncogene is activated. As a less characterized cancer gene, we show CTCF, which is a multifunctional transcriptional regulator, which has been predicted previously as a tumor suppressor, but not really in high throughput studies. We find that it has a number of mutations near its DNA binding domains, which also coincide with the clusters of phosphosites. And the previous literature shows that CTCF differentially binds different genes depending on the phosphorylation status, and there is also evidence that some mutations seem to determine where it's binding, if it's binding proliferation genes or not. And our analysis sort of links to those two ideas together, proposing that phosphorylation-associated mutations may determine how this transcription factor binds. And finally, this is a third gene I wanted to highlight. This is IDH1, which has recognized due to its frequent and recurrent mutation in the blood and brain cancer. And this is a metabolic enzyme. As you can see, there is one very frequent mutation hotspot with nearly 40 mutations. And our model predicts that these 40 mutations somehow disrupt or alter tyrosine phosphorylation, which is nearby the site. And this tyrosine phosphorylation has been so far characterized in one high throughput proteomic screen. It's just a prediction that there may be something going on which relates to the function of this oncogene. As I mentioned, many of these phosphosite-related mutations are quite rare. And in order to bring more understanding, we can integrate them into pathways. And this is how we approach this. This is an enrichment map that shows you many pathways which you would immediately relate to cancer, such as cell cycle signaling or immune signaling, but also other things that are more basic biology and perhaps showing how phosphorylation crosstalks with transcription, for example. Here are some top genes that are highlighted in RNA splicing, and also some top protein complexes are in RNA splicing. I'm going quickly over it because I wanted to focus on something which is a bit novel. It's phosphosite mutations in network rewiring. We know that some kinases recognize specific sites in protein sequence, and this is how they recognize the sites and cause phosphorylation of residues. And we believe that phosphorylation mutations often specifically alter those binding motifs and lead to network rewiring. And this is one example. We know that TP53 is a central tumor suppressor that is regulated by many kinases, including the Aurora kinase. And you can see at the specific site 215, there is an Aurora binding site in which Aurora binds to this R residue here. And the idea of network rewiring comes from a mutation analysis of this site, where we see that multiple Aurora, in multiple samples, the Aurora, the R residue required by Aurora is actually mutated to many other different residues. This is not very frequent in terms of TP53, but it gives us mechanistic insight of how those mutations may lead to differential kinase phosphorylation. And this is a kind of a loss of signaling event. At the same site, we can predict a gain of signaling event where the motif of BDK1 seems to be induced after a specific residue is added to the sequence. So V218 to G seems to give enough binding specificity for this kinase to occur, kinase signaling to occur. There are a large number of such sites that we pull out using our systematic method. So when we look at this number of sites, then we can just say that we will provide more insights into the function of phosphorylation-associated SNVs by considering all those kinase motifs. In total, we find about 1,500 direct phosphosite mutations where the central residue is destroyed. Therefore, no phosphorylation can occur at that site longer. While there is also nearly 4,000 sites where we can predict some sort of kinase network rewiring, which brings us nearly 1/3 of the phosphocyte mutations, we can start predicting the mechanism of how they carry out their function in cancer cells. To summarize the key points, we have a lot of cancer mutations in phosphorylation signaling. They appear to have stronger impact on protein function in general, and this is indicative of driver mechanisms. So when people do mutation analysis, they should consider signaling information as one source of evidence. And then we are talking about active driver, which helps you to distinguish these types of mutations in terms of specific genes. Then the MIMP methods that I briefly talked about helps you analyze those flanking mutations that maybe alter kinase specificity. And in the future, we would like to extend these approaches to other types of post-translational modifications. And also now that we do whole genome sequencing on tumors, we can think how to extend these ideas to gene regulatory motifs, for example. And with that, I would like to thank you and also the people involved in this project. Omar is a grad student in our lab who was building the statistical ideas how to evaluate binding site rewiring, and Gary is an excellent mentor who has guided us through this project, and other people who have provided valuable input. And if you're interested in more information, those studies were very recently published in different journals. So thank you for your attention.
One very quick question. Very nice work. And the other question is not totally related. You look at where the mutation interrupted the phosphorylation site. Is that the opposite thing happening? There's, originally there's no phosphorylation because of mutation. All of a sudden, it has this kinase process. Would it be possible to look at the opposite instead of loss of the process, gain of the phosphorylation process? Thanks.
I'm sorry. Can you repeat the question, please?
Well, you look at the sites there where mutation interrupted it, right?
Yes.
Mutation could do the opposite. It created a site.
Creating sites. Yes. Yeah. There are those cases where we could suggest that sites are created. And I showed you one example here, what has a gain of signaling for BDK1. We are only looking at sites where there's previous evidence that the phosphate group has been attached. So this is a stronger hypothesis. However, it will depend on cellular context, such as we assume that this kinase is expressed and it's co-localized with the substrate protein. If you do it proteome-wide and look at all STY residues and try to predict mutations that create phosphocytes, then you will have a lot of false predictions because there are so many STYs across. So we have done this, but we wouldn't present this as a strong result just quite yet.
Thank you.